Thank you. We are very, very honored to be here. Thank you for inviting us. Thank you for hosting us. We have a fun and very long tutorial for you for the next 4 days. Know that I have brought 3 friends with me, 3 colleagues. 2 of them are in the room. There's Emma over there, and she will introduce herself soon, and Aaron. Emma and Aaron will be joined by Anton. Anton is in the taxi from the airport right now. So hopefully he'll come soon. So when they each speak for the first time, I'll ask them to introduce themselves. I will say we all speak English as our first languages. Sometimes I speak quickly. Aaron speaks quickly. Emma, I think, is slower. If we go too quickly, slow us down. Raise your hand in the other room. Ask questions. At any point in the lectures, please stop us and ask questions. It's okay. Also, during the breaks, you can ask us questions. And we have left time at the end of every day, after the lectures are finished, for people to ask us personal questions if you're embarrassed, or if you have a question about your own research. We will stay around and answer questions too. Okay. Anything else in terms of? Okay, so you are welcome to sit down, and we'll start the lecture now. Yeah. Do you want to go to the other room?
I'll sit in the back of the other room for the first one, just in case.
Okay. Okay. All right. So we have a mixture of lectures for you and hands-on exercises. So now I'll be at the screen here. Okay, so I think it was introduced from the Broad Institute. The Broad Institute was formed as a partnership from MIT in America and Harvard universities. We are now independent, but still an affiliate of both of them. We all work in an entity called the Data Sciences Platform, which is a part of the Broad Institute focused on data sciences and engineering. My background is I started in 2009, so almost 15 years ago, 14, 15 years ago. You'll see the GATK started in 2010. So I started before the GATK. I was hired to build the GATK. So I've been here for a long, long time. I have seen many things. I've seen the technologies in the early days for DNA sequencing, where we had the early Celexa machines from Illumina. We had 454s. We have companies that are no longer exist. And over time, we've seen newer and newer technologies. So for the first time in this workshop, we'll talk to you about long-read sequencing, Pacific Biosciences, and Oxford Nanopore. We have a lot of new modules, new things to teach you since the pandemic. Emma will be teaching you all about structural variation, finding large mutations, which is a very big and complex thing to do. So we have a lot of new material, and I hope you bear with us, and I hope you find it enjoyable. Now, I want to take a poll. I can't see people in the other room, so I'm only going to, unfortunately, be able to see people in this room, but just to get a sense of your experience. And there's no judgment. Any answer is okay. But how many people can use a computer? Okay, raise your hand if you can use a computer. Okay, so we're good. How many people will use Stack Overflow when they do look up something for their work? Few people, though, getting that about half. Okay. How many people know what, VIM is, or Emacs, or any type of, something that's not Microsoft Word? Very few. Okay. All right. So we will try to cater this for everyone. We will teach you a lot of introductory material. So for people who don't know anything about bioinformatics, we will start slowly. We will probably get into details as we go along. Ask questions if you have questions. If you already know everything about DNA sequencing and you're already an expert, some of it might be review, I would say. But hopefully we'll still be able to teach you some things also. Let's get started. The GATK is written, it's software. It does not require any specific operating systems. It does not require really anything. I know this is sponsored by the High Performance Computing Center. We, as you'll see soon, that it's very much focused on being able to be run anywhere, especially in a high performance computing center. We actually have collaboration with IBM and with Intel Corporation to make sure that GATK is optimized for running locally. It's also at the Broad, our IT department is very good. but not world-class like Google and Amazon and Microsoft Azure. So we actually moved all of our processing to the cloud. I'll talk about that in a bit. We're going to ask you all to use the cloud for this workshop. And I'll remind you this again. Oh, you can sit there. It's okay. You can, yeah, it's my stuff. So in past workshops, we've had problems. People brought Macs and PCs. They had Windows, and they had Apples, and they had different operating systems and everything. It was very hard to get everyone to do the same thing and see the same thing. So for this workshop, we're going to ask you to use something called Terra. Hopefully you have all created an account. I'll get to that very soon. But just for the workshop, to make sure you can see what we can see, we're all doing the same thing. We'll ask you to use Terra, which is a cloud-based platform. I'll talk about it in a second. But as soon as the workshop is done, you can go back to using high performance computing. Almost everything we're showing you today is available. Be downloaded and run. There are one or two things that will be hard to do that, but we can still work with you on that too. Okay. And when that last bullet point, reproducibles, we'll make sure that anything, what the point is, again, that we'll show you how we string all the tools together, what the flow is from one tool to the next. And we'll give you the files. We'll show you exactly how those workflows work. This is a little bit of an overview of the timeline. I mentioned GATK was first introduced to the world in 2010. So we're 13 years old now. At this point, this is even an older slide. We are now into version 4 of GATK. We've hopefully I think 4.5. I think when it's coming out very soon. So we'll hopefully continue to keep giving you and providing you software for effort, but at least for the next 13 years too. I will also mention GATK is fully open source. So everything that our group develops, and there are some software we'll show you in this workshop that isn't actually GATK in the same GitHub repository, but it's in the same family as GATK. Everything is open source. We no longer produce any software that isn't fully open source. And that is because we are a not-for-profit institute trying to cure disease. That is the mission of the Broad Institute. And we feel strongly that we are more able to cure disease and help you all cure disease. And as was mentioned, things like COVID, to study that if software is free and open to the world. And we don't judge if software isn't, but ours all is. Actually, this is even an older one. We'll show you a flow from working from raw tissue all the way to the end, so you have your variants or mutations, differences in DNA. We'll give you an overview very soon on that. So we'll show you the whole flow of what happens and how things get processed. We have for cancer and for germline. Germline is non-cancer. For all types of variant classes, for types of mutations, we will show you all of our workflows. We have a lot to go over. And I mentioned everything is published in GitHub, so you can see exactly what we're seeing. It's all public. You can go there and download things and see exactly how to run the workflows, how to run the tools, how to put them together and what things do, and how we run them ourselves. So the same way we run them when we process our data and back in Boston and in the United States, you can run it the exact same way. I mentioned Terra, and again, Terra is a cloud-based platform. So you will all be using Google's cloud for this workshop. Terra will soon also support Microsoft Azure, but not yet, not for this workshop. So hopefully you have no issues there. But again, it's important for you to see exactly what we're doing. Everything, all the data we'll provide for you is already sitting in Google Cloud. And all the workspaces you create, all the pieces, everything you do, all the compute you'll be running will be running in the cloud. Okay. You should all you'll be running on my host object, so my account. So please don't just use it for the workshop, but don't go off and do Bitcoin mining with this account. We'll find out. They'll shut the whole account down. So everyone play nice, please. Okay. All right. So there are many. If you go to Terra, and we will ask you to go to Terra very, very soon, you will find that there's many workspaces, many workflows and tools already prepared for you, so you can see how to run them. And if you want to just run them, you can click buttons. today, when we, and for this week, when we show you different exercises, I want you to try to understand them. Don't just, you could click the buttons and not think. Don't just copy. I want you to think about what they're doing. And I'll ask the instructors to slow down and to show you what they're doing so you understand what's going. But we'll have it all set up so that the data is small. Otherwise, it could take days to process your data. So we have it all set up so things are quick. But try to understand what you're doing. Okay. If you have questions beyond the workshop, we have a very, very large set of tutorials, and we have a user forum where people ask questions and get answers. You can make requests for features. There's documentation for the tools. We put announcements there. So there's lots and lots of details if you want to go to the GATK website. So this is gatk.broadinstitute.org. You can go there. It's open. You're welcome to participate. It's all in English, but that's fine. And we would love to see you there. You can download the latest version of the GATK there. So again, everything is downloadable. I'll repeat again, we're only asking you to use Terra in this workshop to make it easy on us and you. But you can download it and run it on your own laptops. It works well. You can run it in the high performance computing center. Everything hopefully is supported. If you look here, this is our webpage. On the top right, you see you can run it on the cloud and you can run it on HPC. Those give you instructions for how best to set up their high performance computing center for running GATK. And again, we work with IBM and Intel corporations to make sure we have optimized the GATK for their hardware, their configurations. They've done a lot of the work themselves. We have lots of documentations and tutorials. There's a picture of Derek up there. Derek has set up actually a lot of the modules for the GATK workshop, so I thank him. He's not here this time. But there's lots and lots of instructions and tutorials you can find there. So, please feel free to browse and view. The workshop, again, is 4 days. We hope to see you all for all 4 days. We'll start today with some introduction to sequencing, DNA sequencing, to understand it all. And we'll start with germline. Again, germline means inherited from your parents. Tomorrow we'll do some more complicated things further along on the germline side. On Wednesday, we'll start to focus on somatic cancer analysis and the larger CNVs, copy number variation, larger mutations. And on day 4, you have actually on long reads also, I think, MS on Wednesday. Is that Thursday? The long reads is tomorrow. Oh, so you get the special treat tomorrow with long reads then. And then on Thursday, we have kind of the miscellaneous category. So you have single cell genomics, which is a new field in DNA sequencing. There's mitochondria, and I believe that's when the structural variation will also happen. Okay, so I would actually gonna say, so in the email, it should have all been sent out an email where you all signed up for an account, and in it had all these instructions, including a link. Let's see if I can do this. If I can just go down here. So in the pre-workshop instructions, and I'll see this, I think. You have this link right here. Okay, and that has it's a folder in Google Cloud to all of the presentations that we are sharing. So if you want to review anything, you want to see anything, all the presentations are available to you. You could even see tomorrow's if you want. You're cheating and speaking ahead, but it's okay. But you can always review and see things on your own time. Those I will keep those there for a long, long time, at least a year more. I don't plan on removing those. You can copy them. It's all public. Anything is there for you. Okay, and there's also a link to the agenda. The one thing I will say is when you're all finished, one request. I have one favor to ask of everyone, which is at the end of Thursday when we're finished, please do fill out a survey. It's 2 minutes, and you don't have to say nice things. If you say nice things, it's good for us. But we use it to know how we did and to improve for you. Oh, and we have Anton. Anton has come, so we definitely need to clap for that. Yay, Anton! He is great. He has come straight from America here. So please do fill out the survey at the end. I will remind you on Thursday. If you don't make it all 4 days, it's okay, but please still fill out the survey. It helps us help you. This is the 3rd time we're here. we come back for a fourth time, we want to make sure we teach the right thing. Okay, back to this. Oh, Emma, how do I go back? Good enough. I'm gonna stick. Control H. No, good enough though. Okay. Okay, just a little bit about Terra, since you'll be using Terra a lot for the next few days. I want to describe it to you. It was also developed at the Broad Institute. It's developed by actually none of the 4 instructors actively are engineers on that team, but we are colleagues and partners and friends with them. We are, users of that platform. It was developed for the Broad and for the world to run any sort of bioinformatics pipelines on them. Free, open source for the world. The nice thing is, so the U.S. government has certified it to be secure. So you could put secure data on there, which means we're allowed to put human data on there. So you will use human data today. The data we'll be using in this workshop will mostly be public human data. We call them HAT maps, which are freely have data which have use restrictions that allow usage across the world. But Terra in general allows you to set up workflows and share data and run pipelines on the cloud, so you're not limited by the bandwidth. So we don't have a high performance computing center like you all do, and so we would routinely run out of compute in our data center and cry and complain. And so finally we said, forget it. We just went to the cloud and used that today. So you're lucky that you don't have that problem. Okay, so we're going to ask you to do some things now because I tried to get everyone who signed up on our billing account. I want to make sure you're all signed up. Before we start to do the hands-on later, I have to make sure everyone has an account that is working. So I'm going to ask you all to go to Terra. So it's terra.io, and to log in with use the accounts that you signed up with. And I hopefully have added every single one of you in both rooms to the billing account. Again, you're going to be using my accounts. You'll be spending compute and storage on my cost object, which is okay. So I'd like you to make sure you can sign in and confirm you can see everything first. Yeah, so
use the student back to they can use their own computer.
Oh, yes. So how do I, okay.
They can use their own.
So now you can all use your own computers. Yeah. I know you can't.
Maybe you have to open your computer.
Yeah. But can they still see my screen? No. So what I want them to see my screen. Oh, got it. Okay. So now you can all see this.
还没有开机的，开一下脚边的桌机。如果你们自己有带 notebook 的话，可以用自己 notebook 给开机。后面带 notebook 的话可以用这个。WiFi 如果在后面。
Harry, do you want to say that to the other classroom too, or no?
Yeah.
I'm sorry. Okay. So now you'll be able to do things on your computer. And see me here. So for now, I just want you to log in. If anyone has any problems, in this room I have Emma, and just raise your hand and she can help. And in the other room, Aaron, and maybe Anton. Anton's gone already. Be ready. Okay. So as you log in, I want you to make sure that you are on our billing account. Okay. So this tells you what to do. Actually, I changed the name of the billing account, but do this now. So again, if you look on the hamburger icon, you click it, you click on your name. Hi,
I was walking back.
Oh, you're good. You're good.
What did you say?
Oh, nothing. We're good. So I want to make sure you can see billing and groups. I want to see what you're all looking at. All right, so actually don't look for, so just click on your groups and don't look for this one. Is we changed this name for you to be specifically to Taiwan. So I have a GATK workshop in Taiwan. And if you see that, hopefully you can have it. No? Okay, good. So should we look for just for the billing account? Let's have them create let's create a workspace instead. Okay, let's do this. I'm going to ask you to go to the workspaces. So again, go to this menu up here, this three bar menu, and you should be able to click workspaces. And amongst the workspaces, there are featured workspaces. And I want you to find any featured workspace that looks interesting. It doesn't matter, any featured workspace. I would like you to go to that. Don't even do these, any featured workspace. I don't care. Right, so again, just to say, I want you to click on the three bars here, go to workspaces, find featured workspaces, and then find any featured workspace. I don't care which one. And we're going to ask you to clone it. Clone it means to copy it. And the only way you can copy it is if you are on my building project. So let me go to the next screen here. Okay. On that workspace, if you can't get to the workspace, let us know. But once you get to a workspace, any workspace, it doesn't matter. Go to the top right corner with these three dots. And I want you to click clone. Again, clone means copy. You're going to make a copy of the workspace for yourself so you can run it on your own. And you should be able to do that. I think on the next page, I want to keep that up there, but I want to also look at the next page on it.
So I see some people have the billing account and some people don't have it.
Perfect. That's what I want. Well, not perfect, but that's what I want. So if you clone the workspace and you're on the billing account, you should be able to create a workspace name here, any name you want. And you should see the workshop name. I called it GATK Workshop Taiwan, I believe. That makes sense? Okay. And everything else, it doesn't matter. I don't care about the workspace, but I just want to make sure you can all be on this so that later on when we do a real workspace, it works. Good? Okay. So anyone who's not able to clone this workspace and is not on the billing account, yes, so we're going to start adding you. Okay. So the best way to do it is we need your Gmail accounts. So Aaron, Anton, and Emma all have admin access to that. You don't have it? Okay, so Emma will go around and collect names and email them to me, and I will add them, or Aaron, one of us will add them. So again, if you cannot do this, that's okay. We need to add you to the list. So please raise your hand. That's a lot. And we'll come around in both rooms and collect your names so we can add you to our billing account. All right, and adding you is very quick. I just need, this is why we asked you to sign up in advance, and this is why we're practicing it now. Okay, first of all, any questions while we're doing this? If you're not on the billing account, don't worry, we'll fix it quickly. Questions so far?
Go to billing.
Go to billing. Emma will come in and see what we have.
如果你没有那个备用account的，等下就举个手。
I'm just going to go back. I'm going to go back to the previous
如果
page.
正常的话，应该是可以。
So again,
它会出现备用account让你选而已。
you try to do it.
正常的话应该是这样的。
Okay. So again, I'm going to keep going, but just if you don't have access to the billing account, I want you to please keep your hands raised as Emma and Anton and Aaron come around so they can get your name, and we'll add you very, very soon. Okay, before the first exercise, I will make sure I have you all added. Good. That is it for this. I'm going to start now. Emma's control E. Okay. I'm going to now start on the first module. Wait, maybe I'm going to wait 1 second. All right, now I'm going to start. So we're going to actually start learning. So again, no problem. If you don't have access to the billing, no problem. Just keep your hands raised. As we keep going, I'll keep teaching. So I want to keep moving. So the next module is an introduction to DNA sequencing. this is this is not, so this is about the biology of science involved here. It's important for those of you who don't know enough about this to get an understanding of how sequencing works, because it will be applicable when we learn about the processing of that data. So I want you to understand how it's created. So this is, again, everything in this next module is before the GATK. I'm not going to teach you any GATK stuff for the next few minutes. This is just about the sequencing itself. The first thing we need to do is we need to get some. I'm going to keep the presentation in the middle so people can still try to clone workspaces. Okay, so I will be up here for now. Okay, so the first thing we need to do is get DNA or RNA, some sort of biological data. So RNA is floating around in your cells. Remember, you have DNA that gets transcribed into RNA, which gets translated to proteins, the basic tenet of cell biology. So you can actually go from RNA back into DNA, okay, via cDNA. Okay, so you can grab RNA and do RNA sequencing. That's very common to see what has been. So this shows you only the DNA that is expressed by cells. You can take the entire DNA. The important thing, though, is you purify it and you break it into pieces. Even with long-read sequencing, you need to break your DNA into pieces. You cannot sequence 3 billion letters at once. The machines don't work that way. Most of machines are called short-read sequencers. So you can sequence maybe 200, maybe 300 letters, nucleotides, bases, at one time. A long-read sequencers, you can do 8,000, 10,000, maybe 20, 30,000 at one time, but you can't do 3 million. Okay, so you must break up your pieces, your DNA, into fragments. And the fragments are the things that are sequenced. First, you attach adapters to them. The adapters are there useful for PCR. Okay? PCR is just a biological mechanism that allows you to duplicate your DNA. And that's because one piece of DNA is very small. And the sequencer machines use light, they fluoresce light to try to detect it. And so what they need to do is see a little cluster of DNA, not one little piece. So you use PCR to amplify. It's called amplify your DNA. Okay? So now you have little pieces of DNA, all with the same, the same fragment sitting there. And the DNA sequencing machines go ahead and they start to amplify this. So this is an Illumina machine. Illumina is probably the most common sequencer. BGI, I believe, uses a very similar technology. And again, they use these fluorescent glow-in-the-dark dyes on their bases, so you can see them. And all the different bases, A, C, G, or T, have a different color. And so it lights up in a microscope, and all their what is called base calling, detecting this fluorescent lights, is how they detect the bases. The way an Illumina machine works is they have these things called flow cells. So you stick the DNA into flow cells. There's 8 lanes for every flow cell. And your DNA can fall in any of them. And in fact, what most sequencing centers do is they take your one sample and divide it up into many different lanes. And they put many samples on those lanes too. So it's not just my DNA. My DNA is not just on one lane, but it's on all 8 lanes. This way, if there's a problem with one lane, I still get sequencing done on the other 7. And so, but there's too much for just, there's too much availability for just mine. So they put lots of samples all on the same thing. So how do they know the difference between my DNA and your DNA? They attach a barcode to it, a special barcode, just like they do in the supermarket. You put barcodes on things, so they have barcodes on DNA also. Okay. From one sample, so you can then take those barcodes to differentiate and find the reads belonging to your sample, to me. When I'm done, for a normal genome, there are about several 100 million reads just for one sample. And then again, for short read sequencing, there are several 100 million reads, all of which are about 200 to 300 letters, bases long. But that's just nothing. It's just lots of letters right now. So what we have to do is start to process it. So the format for the raw reads, the unprocessed reads, is called FASTQ. If you've heard of a FASTA, that's how you store a reference, which we'll talk about in a minute. The FASTQ format is how we store raw, unprocessed reads that come off the sequencing machine. Okay, We don't need to know all the details, but there are a few things that you should know. So the important thing is there are 4 lines for every read. The first line is a completely usually random or definitely it's a very unique sequence name for that read. So they usually do give a timestamp so you know that it won't be the same as any other sample. So that's there. Then you have the letters. Okay, so here's an example right in the middle here. So there's just some kind of random gibberish that is a unique identifier for that one read. And here's the actual read itself: CCC, PP, CT, etc., etc. And skip the third line, but the fourth line is also important. This also looks like gibberish, like nonsense, but it's very important. These are numbers. Now you'll say to me, wait a second, Eric, this is a semicolon, it's not a number. So this is ASCII representation. So you have to go to the ASCII table and convert it. And these numbers are very important. The numbers tell you the confidence that the machine had in that one base. So this number refers to that base, and this number refers to that. Okay, so 3 does not mean 3. Again, it's the ASCII representation. And so it's very important that that is accurate. So if the machine tells you that this base is 100% accurate, but it's not 100% accurate, we'll have big problems when we go to do variant calling later on to do our analysis, because we're all going to trust that base, but the base is not trustworthy. And if the machine tells us it's 50% accurate, then we'll know to only give a little bit of weight. We will know not to weight it too much. So it's important that these numbers be accurate. And we'll teach you a little bit today. I think Emma will teach you how to make sure we make sure these numbers are very accurate. When we talk about bases, so we talk about the confidences both with these bases and with variant calling. Everything we do is Phred scale. Phred scale is here, PHRP. Okay, so to make it easier to understand, I think we convert all these numbers so they're not probabilities anymore. Instead of saying it's 99.999% confident, we just assign number by taking the -10 log 10 of that number. So let me give you an example. So if I say something is a Q10, okay, then it's -10 times log 10 of that error, which means it's 90% confident. So a Q10, so this is all in log space, okay, exponential. So a Q10 is 90%. Oh, I lost my, oh, here we go. Okay. So Q10 is 90% accurate. So 1 order of magnitude more, a Q20, okay, that's now 99% accurate. A Q30, 1 order of magnitude more, is 99.9% accurate. So already Q30 is pretty accurate. That means there's 1 error in every 1,000. Okay, so most sequencing bases from Illumina are between Q30 and Q40. So that means there are very few errors, 1 in 1,000 or 1 in 10,000. That's very few. That being said, remember, there's hundreds of millions of reads. Okay? Hundreds of millions. So 1 in 1,000 is still a lot of errors. It's a lot of errors. So this is why we still have a job, because it's not so great. Okay, that makes sense? So when we talk about variant calling later, we talk about Q30. Remember, that's 99.9% accurate. And that's usually what an average base is. The accuracy is lower for the long read machines usually, except for with, you'll see on the long read module, Pacific Biosciences has a way to sequence the same piece over and over and over again, so they improve the accuracy. All right. So for those of you who don't know, they have things called whole genome sequencing and whole exome sequencing. So the genome just means we sequence hopefully every base in your genome. You have about 3 billion bases. So we're sequencing 3 billion. And we say something is 30x, that means approximately, on average, at every position in the genome, there are about 30 reads covering it. Okay? So again, when you've broken up your DNA pieces, you're sequencing multiple copies of your DNA. And on average, across your entire genome, you see 30 copies of the DNA at any position. That's very expensive. So the costs have come down. Now a whole genome for Illumina is about $600, $500. They say it's $100. They're lying. It's not $100, not even close. I don't know about BGI these days, but talking about $300-400 for a genome. It used to be much more expensive. It used to be $1,000 or $1,200. The PacBio machines, Specific Biosciences in Oxford Nanopore, are more expensive. They are now $1,500. They used to be $60,000 US dollars to sequence one sample. So it's a lot more expensive. So if you're in a clinical hospital trying to find a disease that's causing, that's hurting a baby, you want to make sure you cover everything and make sure you know exactly what's happening. But when you're doing a large research project where it's more important to have more people and maybe a little bit less accuracy, what many people do is use whole exome sequencing. The exome are just the exons. Okay, so it's just the genes. So just to remind you, most of your genome, about 99% of your genome, is not coding. Okay? It's not exons, not your genes. We don't know exactly what they do. There are many theories from they do nothing, they're junk, to they're very important. They promote all the transcription that happens. So we won't go into that today, but know that an exon, an exome, is about 1% to 2% of the genome. And what they do is they only sequence that much, so it's a lot cheaper. It's not 1/100th the cost, but it's a lot cheaper. So an exome today is under $100. So you can sequence all the genes, and which is important because most of the mutations that are relevant and matter to medicine are in your genes. Right? So that's why people do exome sequencing. And you see in this picture, you will not get any coverage across the rest of the genome though. So these stars represent mutations. And you'll see, and the red bars are the sequencing reads. And you don't get any reads there, but that's okay. You're doing that as a trade-off for cost. There are also these things called panels. These are arrays, also called, I don't know what they have in Taiwan, but in America we have 23andMe and Ancestry.com to see, to find out what your ancestry is. So those things really cost like $15, but they'll charge you $100 on, online. They're a lot cheaper, but you only get a few places in the genome. When I say a few, you get a few 100,000. That's a lot smaller than the exome, which is about 1% of the genome. Is it too loud?
It's just this.
Okay, fine. I'm going to hold the mic further away. Thank you. Okay, so now you can see this in actual picture. So this is very important. This tool that I'm showing you here is called IGV. And you will all use IGV. If you haven't used it yet, get excited because you will soon use it. IGV is called the Integrated Genome Viewer. It was partially created for me many, many years ago, back in 2009. I was trying to figure out how to find insertions and deletions, and it was impossible. And so the guy next door to me in my office said, "Oh, I'm trying to create this viewer. Let's work together." And so we worked on, so he did all the coding, and I said, "No, this is not working. This is working." I'm very old. Okay, so what you can see here are the actual reads. Again, these are the pieces that are read off from the sequencer. Okay, that's what each of these bars is. So one bar is one read of DNA. Now a fragment is read from both sides. You usually get 2 reads from one fragment. These eyes here represent insertions. You can't see them. If you hover over them in IGV, it'll pop up a little window and tell you what the sequence is in there. Anytime there's a difference so the reference sequence is here on the bottom. Everything is color coded. So A's are always blue. C's are always blue. A's are always green. G's are always brownish orange, and T's are always red. So what this means here, for example, is that there's a T when the reference is not a T. If the base matches the reference, there'll be no color. It'll be red. And so the fact that you have a whole line of insertions here means that there's probably something real. It's not one of those 1 in 1,000 errors. But the fact that there's one little blue here and nothing else down there means that in this position of the genome, there's just one difference. It's probably an error. And we know errors happen occasionally. Okay. This track here shows you coverage up top, and you'll see if there's enough of the difference here, it'll show it up here. So what this is showing here is that there's a red and an orange. So there are many orange differences here And you can't, you'll scroll down, you can see the red ones too. So IGV is very important for viewing the reads to understand what's going on. Good questions? And notice also that this is an exome versus a genome. So these big blue bars on the bottom are this is an exon, and this is not an exon. So there's no coverage here. You do see coverage outside of the exon a little bit. So when we do variant calling on an exome, the exome is all the exons. You can do calling across the exon and for about 20 bases on the sides. Okay, so you do see some regions outside. Yeah.
How do those reads highlight in red mean?
Great question. I will show you an example of many of them. So the ones highlighted in red are ones that are messed up usually. And they're messed up because so again, this is very important. The reads are usually paired end with Illumina, the short reads, which means again, I'm just going to go back to answer that question. Okay, so you have this fragment. When you do the sequencing, you sequence this end and that end. So you have two copies, and you don't read all the way through. The fragments are about 400 bases long, and you read about 150 on this side and 150 on that side. Okay, so you have about 300 bases, but not consecutive. 150 from this side and 150 from that side. You don't want them to overlap because then you're wasting sequencing. Okay, and so now I'm going to go back to the other side. So again, you expect, let me say, this end to be about 400 bases from this end, because usually these are about 400 bases long. From this end to that end is 400. So you expect 400 bases difference. This is telling me that the other half of it is further than 400 bases. Okay, something is wrong with the pair. The other one is farther along, too farther along. Just because they're red doesn't mean that they're okay? Or they're too close. Something is too close. Something is messed up about this pair. That's what the colors mean. And I'll show you some examples of when you see things go wrong. Good. You'll also see some directionality. There's a little arrow here. This means that it's that end, and an arrow the other way. That's fine. Here, there's an arrow this way, saying that that's from the other side, reading in that way. Okay, so different exome kits create exomes form differently. So you'll see these are just examples. Some of them you can see kit B on the bottom is a lot better in this region here, and not so much over this gene here. But kit A does a lot better there. So it's all hit or miss depending on the kit, and that's nothing we can do about that. That's not something we can control. Bless you. So these are the things that can go wrong. This is why understanding, so a lot of the processing that GATK does is to handle errors in the sequencing data that aren't our fault. So Illumina would tell you our data is perfect. It's not perfect. All right, so things that can go wrong, and I have them listed here. I won't just read to you, but that second, and I'll show you some examples, is you can have poor coverage, you can have high chimerism. Chimerism is what I was saying before. It's when the two reads in a pair are messed up somehow. They're usually too far apart. Usually one maps on chromosome 1 and the other one's on chromosome 2. Well, that doesn't make any sense. Right? They should be 400 bases away, not different chromosomes. So the only way that makes sense biologically is that there's a translocation where the two chromosomes have merged, which happens, of course, but it's unlikely. Insert size, again, so when I said that the fragments are 400, I expect the ends to be 400 bases apart. So we have a distribution to see how long these fragments are, and that's called the insert size distribution. Insert size is the length of the fragment. And so these should all look at. I'm going to show you pictures of all this stuff soon. Okay, other things. So that's true for everything, for exomes and for genomes. For exomes, you also have other artifacts that are specific to exomes. There are times we'll see problems in exomes that don't exist in the genome of the same exome. Okay, and usually it's because the coverage is uneven. And some exons capture better than other exons. That's a problem. And you'll see later on when we talk about copy number variants and detecting them, it's very hard. Statistically, it's very hard because different exons have different depths, and you're trying to figure out which one has a copy difference. That's hard. So there's a lot of math involved. And then with whole genomes, it's much easier to get contamination in there. The exomes, because the capture process itself selects just for the exomes, you do get a lot of removal of contamination from other organisms. But for genomes, we all the time. So we, at the Broad, for example, in our sequencing center, we sequence saliva, spit. And sometimes they're from sick kids, and kids do not read instructions. They don't. I have 5 kids. I know they do not read instructions ever. So they don't know to wash their mouths out well. And so all the time we see cow DNA, hamburger, chicken DNA, pig DNA, all the time. At first, we could not figure out why. I'll show you an example of what it looks like when you have that contamination. Turns out there's lots. It's very easy to get that inside. And again, with exome, that doesn't make it through the capture. With the genome, it shows up all the time. Okay, so the other thing to show you, and I'll give you a lot of examples now. So the part of the genome that you're in also matters. So there are parts of the genome that are called GC-rich regions, which are many of the bases are G's and C's, and those are harder to sequence. They just are. There are a lot more errors in those machines. Okay, so this is an example of uneven coverage. We told you for genomes. So this is a genome here. Okay. But there are lots of parts all over that just don't sequence well. And so, and it's very inconsistent depending on how you see things and the technology you're using. Okay, so here's an example of an exome versus a genome. Just look at how, even if you look at the genome, okay, you can see that the coverage here is different as you go along. This is the way it is. Now, with exomes, okay, it's just distribution. So it's also very possible. So we have this thing called mean coverage. The mean coverage is, again, the average coverage across your whole genome. So when I say the mean coverage is 31.6, that means on average, over the 3 billion bases, there are usually about 31.6 reads in a neat position. But that could be very, very variable. It could be, in one part of the genome, there could be billions of reads and nothing the rest. It's just an average. So we also have measurements for percentage of bases at least 15x. 15x means they at least have 15x coverage. So I know that will detect issues where the average is just a bad average. Okay. Sometimes there are regroups that have a lot high coverage and some that don't. And sometimes that's okay. Just because a regroup, again, a regroup, it's very important, is the combination of a lane and a sample. So I told you that in my example before, my sample was sequenced across 8 different lanes. So I have 8 different regroups that are all comprised of my DNA. Okay. So what will happen sometimes is, a customer will say, I want to buy a genome at 30x coverage. And let's say they sequence and I'm only at 28x coverage. So they don't throw it out, they sequence 2 more x. And that's very small. So it's okay, it's normal to top off. It's called a top off of the sample to get there. But sometimes low coverage, if you're trying to get high coverage, is not good. So everything depends on the situation. And all that is part of the QC. So this question before was about why there are colors in IGV. So this is an example of a very bad sample. Very, very bad. You see lots of color. Lots of reads are messed up. And again, for each one of them, their mate, the other half of it from the other side, is aligned on the reference somewhere else, some far, far away. You'd expect it to be about 400 bases away, and it's much more than 400. And I believe when Emma talks about structural variation, she'll talk a lot about this. This is very important to me. So we call these chimeras. You expect to have less than 1%. You expect to have some. Okay, nothing's perfect, but not more than a few percent. This is an example of bacterial contamination, or cow DNA or chicken DNA. This is what you see. Okay, so again, the insert size is the average fragment size, and the fragmentation actually happens by sound. Okay, it's all audio, and it's not perfect. There's a distribution, but it's usually around 400, and then something lower. But notice the peak here is very small. And so that shows up here where you have reads, and all of a sudden you have just a ton of reads, and this scrolls down thousands and thousands of reads for one spot. And all of these colored here are soft clips. And again, Emma will teach you all about soft clips in her module, but there are parts that don't align. So what's happening is the read is partially aligning to the human reference and partially not. And it's because it's bacteria genome and the processing doesn't know where to put it. They think it's a human because it's mixed in with the human DNA. So they put it in the spot where it's closest, even though it's really bad. And so you have piles and piles of these bad reads with lots of color here, a mismatch at one spot. And so so we don't throw out the sample, but it's important for us to filter out these reads and know that they're a problem. All of this is about the QC. So there's a lot of QC that we look at before we are even ready for processing. A lot of that. And again, it doesn't mean we throw out a sample if it fails QC, but we go back and we process it, we remove reads, we add coverage until we're ready to proceed. Questions so far? I don't know if there are questions in the other room, but they should feel free to jump in and let me know. I don't know if there's a microphone next door. Okay, who knows? Yes, good, thumbs up. Okay. So I am done here. And at this point, I'm going to hand it over. Wait, on the agenda, are we good? Or do we have a break? Someone should keep me honest here.
I think we go straight into pre-processing. It's a break afterwards.
Okay, so we're going to go into pre-processing. And is this Emma? Yeah. I'll remind you, Emma, to introduce yourself. Control H. Okay, so I hand the mic over.
All right, thank you. I want to make sure that everyone in this room can hear me and in the other room. So in this room, if you can hear me well, can you give me if you can't hear me, can you raise your hand? Okay, and then if there's any issues in the other room, I trust you will find some way to communicate that to me. One other thing before I get started is if you talk to me or Anton or Aaron about not being on the billing project, I added everyone whose email we got. So if you can go check again if you are on that billing project. If you continue to have an issue, you can raise your hand and talk to one of the other presenters. All right. So we've just talked.
I'm not sure I'm here for that.
Oh,
Okay. So I'm gonna proceed, but if you have any issues with the billing project, Eric can help you out in this room and in the other room, Anton or Aaron can help out. Okay. Sure. So we just talked about how we perform sequencing, and the next step in the process is data pre-processing. So what this means is all of the data just came off of the sequencer, and we want to get it ready to perform variant calling. So the 3 steps of this process that we'll talk about right now are mapping. So we need to get the reads aligned. We need to get the reads aligned to the reference genome so that we know actually which part of the genome we're talking about. Marking duplicates and base quality score recalibration. So just as an overview, this kind of stage of the process is all about, taking in this huge pile of reads, so sequenced fragments of DNA, and making sure they have mapping information and correcting for various technical issues that might have come from the library prep or the sequencing process itself, including artificial duplicates and then base quality score recalibration. So we're trying to clean up our reads before we use them for variant calling. So step 1 is to align the reads back to the reference genome. And the algorithm that we use to do that for DNA is called BWA. And that stands for the Burroughs-Wheeler alignment algorithm. And actually for For exomes, we use BWA, and for genomes, we use BWA-MEM, which stands for maximal exact match. That refers to the first step of the alignment algorithm, is to find the place in the reference genome where the sequencing fragment that we're looking at has the longest exact match with the reference. Okay. So what we want to do is look at our sample genome and figure out how it corresponds to the reference genome, because our reference genome has a lot of annotations. It can help us understand what we're actually looking at. But there's a couple of challenges with that. So shown here, we have our reference genome up top, and then we have our sample genome on the bottom. And you can see that the sample genome has some differences from the reference genome. So there is, the blue region is a little bit shorter in the sample than the reference. There's actually 2 copies of that green region, and there's several stars which indicate single nucleotide variants. So the aligner has to be able to handle mapping the reads while kind of working around all of those differences that are in the sample and the reference, because we want to be able to discover those differences later during variant calling. But right now, we just want to get those reads mapped back to the reference. And the other challenge at this step is that we're only looking at basically one read at a time. So we only have a approximately 150 base pair sequence. It would be a lot easier to do this if we had the whole genome in one piece, but actually we have very short fragments that we have to handle. So we're kind of mapping each read one by one, and for many reads that goes pretty easily because most of the genome is quite unique. So if you have 150 bases, you can find one good location in the reference. You can find one good location in the reference where that read aligns very well. But sometimes there are regions in the reference genome that look very similar to each other. So for example, the green region in the reference, there's 2 of those in the bottom image. So that is indicating 2 regions in the reference that are essentially duplicated. They're very similar to each other. So if you have a read that looks like one of the green regions, how can you tell which green region it actually aligns back to? And part of the answer is the paired end Illumina reads. Those are really helpful in this situation because not only can we look at where one read aligns to, but we can also look at the alignment of its mate. So remember, as Eric went over with paired end sequencing, we sequence from both ends of a fragment. And so we know that those should be approximately 400 bases apart from each other because we know the approximate length of those sequencing fragments. So the position of the mate of a read can be a tiebreaker. And let's say in this example, if the mate of the green read aligns to region 1, the blue region, then that helps us know that the green read is in region 2A instead of 2B here. Okay, so when we have aligned our reads to the reference genome, let's talk about how that information is stored in the file format. So we use a sequence alignment map or binary alignment map to do this. These are basically the same format, but one of them is compressed. So let's talk about what that file looks like. We start with a header. So all of the lines that start with the @ symbol are part of the header. And all of those lines contain metadata that applies to all of the reads that are in the file. So that includes information about the read groups. So remember that was each sample or library can be spread across multiple lanes of the sequencer, and each of those combinations of a sample and lane is a read group. And we store that metadata in the band file. Following the header, there are the records. So there's one record or line for each read. And those records contain structured information about the read. So let's go through each part of the record one by one. We start with the name of the read, and that often includes metadata about the flow cell name or coordinates. Next is flags. So this field contains binary information about the read. So questions like, is the read paired? Has it been mapped? Is it duplicated? So all of these are yes or no questions. And then each of those gets a 0 or 1 in binary. And then the combination of all of those binary fields is converted to an integer. So that's why you see 99 in the fields for flags instead of a bunch of 0s and 1s. It's a way of compressing that information. Next is the position. So we've mapped this read to a position on chromosome 1, followed by the mapping quality. So how well did that read actually align to that position on chromosome 1? And the mapping quality is Fred scaled. So that means that it is equal to -10 log 10 of the probability that the read was mapped wrong. And so 60, shown in this example, is actually the highest mapping quality that you'll see, and that corresponds to a 1 in a million chance that the read was mapped wrong. The next field is the cigar shade, which contains a high-level representation of how well that read aligned to that position. And it's a little complicated, so we'll actually talk about it in the next slide. The next field is information about the mate of that read. So for example, the equal sign that you see indicates that the mate of this read was mapped to the same chromosome as this particular read. Well, next is the actual sequence of bases for the read, followed by the base quality scores in Fred scale again. So for each base, how confident were we that that was the right base call off the sequencer? And the final field is metadata about the read. For example, the read group, any alternate alignments. And you'll see over the course of this presentation that we really like to keep the metadata about the reads with the information about the reads themselves. So you may have had an experience with just having to wrangle a ton of different files. This is kind of our attempt to reduce the number of files that we have to work with and keep all of the metadata and the data in the same file. And we care about keeping the metadata around because there can be systematic biases in the data that correspond to, say, the read group. And we want to be able to go back to that metadata and detect those issues later. So I promised we would talk about the cigar strain next, and here we are. So cigar stands for Concise Idiosyncratic Gap Alignment Report. Long name, doesn't really matter. And as I said, it summarizes the structure of the alignment. How well does this read align back to the reference in the position it was mapped to? So let's go through each of the components that we see in this example. We start with 1S. So that means that one base at the beginning of this read was soft clipped. And soft clipping happens when there is some sort of issue at the start or end of a read, and the start or end doesn't align back to that position in the reference. And when that base is soft clipped, we actually don't include it when we encode the position of the read. So this position of 2, where this read was mapped, refers to the second base, the C in this example. Okay. So after the soft clip base, we have 3M. So that means 3 bases were an alignment match. And it's important to note that that doesn't mean that we found the exact same bases. It just means that structurally there was a match between the read and the reference. So if we skip ahead to the next example with an M, so there's a 2M, and you'll see that there's actually a mismatch between the sample and the reference. There's a C on the read where there's a T in the reference, but we still consider that a match because it aligns structurally. And the variant colors will go back and detect that there was a substitution later on. The aligner is not, that's not the aligner's job to detect the substitutions. Okay, and the other symbols that we see here are 1D and 1I. So there's a single base deleted relative to the reference, and there's a single base inserted relative to the reference. And there are a couple of other operators that appear in a cigar strain. These are kind of the most common, but you might encounter a few others in a given band. Right. Okay, so when we're performing alignment, typically the file format that we get from the sequencer is called a FASTQ. And then we use BWA mem for whole genomes to perform alignment and turn that into a mapped band file. However, the FASTQ file from the sequencer doesn't contain all of the read metadata that I was talking about earlier. And we really want to keep all of the read metadata. So how do we get around that? We get an unmapped BAM from the sequencer instead. And the unmapped BAM is just a BAM file, but without the mapping information. And that file format can contain metadata about the reads. Then we convert it on the fly to a FASTQ in order to run BWA mem to perform alignment. But then we can use a tool called merge BAM alignment to combine the metadata in the unmapped BAM and the mapping information in the mapped BAM to get our final product, which is a BAM file that contains both mapping information and metadata. So I've been really focused on DNA so far, but I just want to note that things look a little bit different when you're dealing with RNA sequencing data. So just as a quick refresher, for RNA, we are sequencing the messenger RNA after the introns have already been spliced out. So if we go to align that RNA back to the reference genome, we'll encounter big gaps wherever there was an intron in the reference genome that got removed before we ever sequenced the RNA. So in the cigar string, that will appear as N10, which indicates a very large gap in the alignment back to the reference. And so clearly the RNA data behaves a little bit differently in terms of alignment. So we actually use a different tool. We don't use BWA. We use the star aligner, which can handle all these intron junctions. Okay, so now we have aligned our reads to the reference genome, and we have our mapped BAM file with all of the metadata. And the next step in the data pre-processing pipeline is to mark duplicates. So we assume that every read is corresponds to one independent sequencing fragment. So we assume that we only read each sequencing fragment one time, and each read is kind of independent in that way. And that's an assumption that needs to hold in order for our variant callers to work. Because if we sequenced a single sequencing fragment multiple times, and let's say there was an error in that sequencing fragment, then that one error would show up multiple times. And then the variant caller would look at that and say, okay, well, that looks like a real variant. We see it in multiple reads. There's a lot of evidence for that variant. So that could result in a false variant call or an incorrect allele balance. So this assumption that we have one read for one sequencing fragment is very important. And so we want to find duplicates where we read a sequencing fragment multiple times and remove them so that we can perform variant calling correctly. So where do these duplicates come from? There are kind of two places. One is library duplicates. So if you used PCR for amplification in your library prep, PCR creates many copies of each sequencing fragment. And that means that you've increased the probability that you're going to sequence each fragment. But you're also probably going to sequence a lot of the copies. So you will end up with duplicates from PCR. The second type of duplicates can occur even if you didn't do PCR because it happens during sequencing. And those are optical duplicates. So this is an image from a sequencer showing, what is the sequencer actually seeing. And each of those little fluorescent dots are a cluster on the sequencing flow cell. And each dot should correspond to one cluster, which should correspond to one sequencing fragment. But sometimes things go a little bit wrong. Maybe you over-amplify that cluster, and the sequencer reads that as two clusters. And then you can end up with an optical duplicate. Okay, so we know that we have these duplicates. How do we go about identifying them in the sequencing data so that we can ignore them? So because of the way the sequencing fragments are created, it's very unlikely to just by chance end up with two independently created sequencing fragments that have the exact same start position or end position. Right, they should kind of all be in different places. So if we look at our reads and we see that a lot of them have the exact same start position, we actually assume that those are duplicates because that shouldn't happen by chance. So we look at the unclipped start position. So we ignore any soft clipping and then look at the start position of the reads. And if we see multiple reads that have the same start position, we consider those to be duplicates. And if you have paired sequencing data, then we actually look at the 5' end of both mates, because it's very unlikely that you'll have the same start position. For a single read, it's even more unlikely that you have the same 5' position. So start of one mate and end of the other mate. It's even more unlikely that those positions would both be the same on both of the mates. So for paired reads, we check both. And if we do find duplicate reads, then we will choose a representative read based on the base quality or other criteria, and we'll mark the other ones as being duplicates. And that duplicate status goes in the flag field of the SAM file or GATK file, because it's one of those binary questions, yes or no, is it a duplicate? And in that way, we just mark or flag the duplicates. We actually don't remove them from the BAM file, but downstream tools, including most GATK tools, will just ignore duplicate reads by default in order to have the correct behavior. Okay, so we have aligned our reads back to the reference genome, and we have marked duplicates. The next step is to recalibrate our base quality scores. So in terms of the bases that come off of the sequencer, they're not always going to be 100% correct. There will be some errors, but each base comes with its own quality score to tell us how confident we are that that base is correctly called. So it's okay if there are a few errors in sequencing, as long as we can tell that those errors have lower confidence. We can treat those correctly during variant calling. But the quality scores that are given to us by the sequencer are not always perfectly calibrated, because there can be systematic biases that arise, during library prep, during sequencing, that mean that those base quality scores that come off of the sequencer are not quite right. And we want to make sure that the quality scores are an accurate reflection of the base quality so we can get our variant calling right. So why are those errors? Where could those arise? For example, remember that a single sample or library can be split across multiple lanes of a sequencer. So maybe there was an issue with just one lane. So just one read group has an issue. And if we observe that there's a systematic issue with just one read group, then we can actually correct for that. So that's what happens during base quality score recalibration. So to kind of illustrate the problem a little bit, we have a couple of plots. Looking at the left-hand plot, we have calculated empirical base quality scores based on a gold standard truth set of known variants. So we actually know how accurate the base qualities are for this set of variants. And that's on the y-axis, and the reported quality that comes off of the sequencer is on the x-axis. And the original base quality scores are plotted in pink. And if the original base quality scores from the sequencer were a perfect representation of the base quality, we would expect them to lie on the x equals y line, because the empirical quality and the reported quality should be exactly the same. But you can see that there's actually some deviation from that x equals y line. So after recalibration, we plotted those recalibrated scores in blue, and we now see that they lie along the x equals y line. So those scores are a good reflection of the empirical real base quality. And the right-hand plot is kind of zeroing in on one of the factors that can systematically bias the base quality scores, which is dinucleotide context. So this means that the base quality of a particular base is actually influenced by the identity of the base that came directly before it. So the sequencer does better or worse on base calling depending on the base that came before. So in this plot, if our empirical and reported scores were exactly the same, then we would expect the line for each of the dinucleotide contexts to be at 0, because we're plotting the difference between the empirical and reported qualities. But we see in pink that the scores from the sequencer are definitely not at 0. There's a lot of variability. So, for example, if you look at the CG dinucleotide difference in quality scores, we see that the sequencer is actually overestimating the quality of the G in that CG dinucleotide context. So after recalibration, we bring that line in blue back to 0, and we've corrected for that source of bias. Okay, so hopefully I've convinced you that this is a necessary step. So let's talk about how we actually do it. So we know that there are these sources of systematic bias that influence base qualities, and those include the read group, as we mentioned, the dinucleotide context, we already talked about that, the reported score from the sequencer, and the machine cycle. So how far from the beginning of the read are we? And so for each kind of combination of all of those covariates, we will count the number of mismatches from the reference over the total number of bases. But we ignore known variant sites, because what we're trying to do is approximate the number of mistakes that are present in all of the reads in that category, that combination of covariates, so that we can estimate how badly did we do for that specific combination of factors. And we can use that estimate of the number of mistakes to recalibrate the base qualities for the reads that have that combination of covariates. So in terms of those known variant sites, we use population databases, and we look at the common variants that we've already discovered in a whole bunch of other samples. But let's say you're not working with human data and you're maybe sequencing a species that doesn't have a database of common variation. What would you do to populate those known variant sites? And the answer is bootstrapping. So what does that mean? You can perform variant calling with your original quality scores, use those variant sites that were called as the known variant sites for BQSR, then perform base quality score recalibration, perform variant calling again, and kind of keep repeating that process until you converge on a stable set of variants, and then you know that you're doing pretty well. So looking back at our SAM file, how do these base qualities actually appear? Showing the recalibrated base qualities in blue and the original base qualities in pink. And it kind of just looks like a key smash, like I just like let my cat walk across the keyboard. The reason for that is that these are Fred scaled base qualities, and then each base quality is converted to its ASCII representation. So instead of an integer, we get an ASCII character that corresponds to that integer. It's just a way of compressing the data. And I will note that there's room in the file to store both the recalibrated and the original base qualities. But at the Broad, we actually don't keep the original base qualities anymore. We found that the recalibrated ones were good, and we just don't really need to go back to the original ones very often. So we don't keep them to reduce the amount of storage that we have. Okay, so we got our data off the sequencer. We aligned all of the reads back to the reference genome. We marked duplicates so that we can ignore them downstream. And we performed base qualities for recalibration. And after this whole data preprocessing pipeline, we are now ready to send our data to variant calling, which we will cover later today. So that's all for preprocessing. Any questions on that? Yes.
I wanted to ask, how do you store the data in the Working Institute? Because you have Pascal, you have another band, especially there is a band called Realign, Intel Realign band, and the VCF. How do you store? You store every band, or only store part of?
Okay. So the question is, there are all these different file formats at different stages. There's the FASTQ, the BAM. Which one do we, how do we store that data at the Broad Institute? And typically we compress the BAM files and use the CRAM format so that they're a lot smaller. And then we'll store those instead of the BAM file. But you can always go back to the BAM file from the CRAM file. You can even go back to the FASTQ. So what we really keep is the CRAM file and later the joint call VCF. Good question. Any other questions? No. Okay.
And I have one. You know, do you trim before alignment?
Do we trim the reads?
Yeah, you know, you start low quality reads or trim certain lengths of the reads. Do you do that?
No. Yeah, so we don't discard low quality reads or trim them before performing alignment.
The only thing that's removed is the GATK sequence.
Right.
Or you know, those adapters.
Right. So Eric is adding that what we do remove is the adapter sequence because that's not part of the actual read. It was just to help during the sequencing process.
We have a question in another room.
Okay.
The question about the modification process. So as we know, during library preparation, some people use a Omicron-based method for library preparation. So in that kind of method, the reads come from the same amplicon will all have the same start site.
Sorry, the audio quality is kind of bad. I can't actually hear. Maybe we can just have you come into this room. Okay.
A bit more on.
Other questions in this room?
Yeah, all right.
Oh, okay.
Yeah.
Thank you. So I have a question about the mock duplicating process. So when we use the library preparation, sometimes we use empirical-based method. So in that kind of method, all the reads come from the same empirical. We have the same start side. So how do you deal with this kind of library preparation protocol during the market degree process?
So I'll let Eric take that.
Okay.
So it's a good question. So this is a specialized type of capture where you've designed a specific probe to get a certain speed place in the genome. So all the reads start from the exact same spot. Yeah, so you can't remove duplicates that way.
Okay.
It's, all the processing still happens, but you can't remove that because every read starts at the exact same position. So in that case, there are potentially other ways of detecting duplicates based on the exact same error profiles, but for the most part, you wouldn't remove duplicates then. Yeah.
So can we skip that?
You would have to skip that. Yeah,
Okay.
yeah. Definitely don't, because you'll remove everything.
All the reads. Okay. Thank you so much.
Thanks for coming over here. Sorry.
Sorry, I just have a really small question. Like in the part where we explain cigar, CIGR.
Yeah.
Like can you explain again like what S stands for? Like it's a mismatch or it's not a mismatch? And in the part I see like a mismatch in the red block. But what's that not inside the reports? So
Okay, let me go back to that slide.
Or do you want to go to that's the slides to show the unmapped the artifact slide up?
I don't know where exactly to find that, but maybe start
like
here.
the seventh position, and why is that not in the reports?
Yeah, okay. So the first question was to go over what the S stands for again. So that refers to soft clipping. So basically some portion of the start or end of the read didn't align to the reference at the location that the rest of the read mapped to. So that would show up in IGB as that like rainbow of mismatches. So basically it's a mismatch, but specifically at the start or end of the read. And then the second question was about the T and the C that are different. Was that it?
Yeah.
Okay. Yeah, so those are still included as an M or match in the cigar string because the structure of the read matches at that position. So there wasn't an insertion or a deletion. The read can still align back to the reference structurally, but there is a sequence mismatch. The base is not exactly the same, but we can still align the read there, and then the variant colors will pick up on the sequence mismatch. The aligner just isn't kind of designed to handle that. It focuses more on the structural alignment.
Thank you. Thank you.
Thank you.
Can I say something?
Okay.
That's a great answer. For the soft clips, the most common there are two causes usually for soft clips. One is this: the read doesn't match because there's a structural variant, which Emma will show you on Thursday. But another common thing is that with the sequencing itself, the sequencing machines will often, try to sequence 300 bases, but at the end it does a bad job, and the sequencing machine knows it's doing a bad job. So instead of telling you the confidence is 50%, it tells you the confidence is non-mathematical. It chooses Q2. So in the ASCII, it's a pound sign, a hash sign, and Q2 actually mathematically doesn't mean anything in Fed. So they assign bases where they just have given up to Q2, and so you often see the soft clips. The ends of the reads are all these very low quality bases that the aligner doesn't use at all. So usually soft clips like these Q2 bases at the end of the reads. Other questions?
Next question. Come up here so the other group can hear.
Thank you. Quick question. Can you go to page 13 about RNA-seq?
Okay.
Yeah. So You mentioned there is a major difference from RNA-seq with the cigar strings by definition of the tools. Can you reiterate the difference between RNA-seq and DNA and how they're actually different? Do we use a different recalibration algorithm or something? Can you be more specific? That really didn't catch it. Thank you.
Okay. Yeah, so I'll kind of go over again what are the differences that I talked about here, and then if there was anything specific, I can ask about that. Yeah, so when we're doing DNA sequencing, we are, well, if it's whole genome sequencing, we are sequencing all of the bases, intron, exon, intergenic, it doesn't matter. So when we go to align those back to the reference, they can just align to the reference genome, which was built from DNA and contains the exons, the introns, the intergenic sequence, all of that. But for RNA sequencing, when the RNA is transcribed, then the introns are spliced out. So there are junctions where one exon is right next to another exon, but in the reference genome, there was an intron in between. So those portions of the RNA sequencing data don't match the reference genome because they're missing those introns. There's a new adjacency between those two exons that used to be separated by an intron. So when we're performing alignment, the aligner has to be able to handle that and align kind of both sides of the read, to exon 1 and exon 2, while knowing that there's actually a pretty big gap compared to the reference that used to be an intron. So in the cigar string, that shows up as N10, that indicates a big gap. So remember, the cigar string is summarizing the structural alignment, and that's a big difference in structure from the reference genome. And the BWA aligner is not designed to handle that. It expects genome sequencing data, but the STAR aligner is built for RNA sequencing data. So that's what we use to handle all of these junctions where there used to be an intron in the RNA data. Does that answer your question?
Yeah.
Okay, awesome. Thank you. Okay. Yes.
Suppose you don't trim the bad quality rates, then how would happen if you don't do the best recalibration?
So the question is, what would happen if you didn't do base quality score recalibration?
Yeah, because you don't trim bad quality rates, right?
Right, we don't remove bad quality reads before alignment. I'm not sure I entirely understand the question.
So since you don't trim those bad quality rates, then it doesn't matter that you don't do the recalibration of the base quality, right? I mean, what will be affected if you don't do the recalibration?
Okay, so
Maybe put that to the how that part. Because I'll describe it as.
okay, so Eric is saying we'll go over that in more detail during faculty
Is that okay?
color.
Yeah, sure.
Yeah, we'll describe it.
Okay.
So let's take a 10-minute break.
Okay.